All right, good morning, everyone. My name is McCain Vogel, and I'm assistant editor at No-Till Farmer. So thanks for joining us today for the live product demonstration webinar titled Managing Crop Residue, Improving Soil Microbes, sponsored by Direct Enterprises. So just to let you guys know, this presentation is going to feature DEI co-founder of Plant Earth Agronomy, Mitchell Feeney, as he provides key answers and alternatives to a variety of seed and soil health questions. A couple housekeeping items before we get started. So as a reminder, there's a Q&A control panel on the bottom of the screen. You should see it there, and you can feel free to use that to submit questions anytime throughout the presentation, and then we'll leave time at the end to go through as many of those questions as we can. And just another reminder, we're going to record today's session, so in the event you want to go back and watch it or share it with anyone else that wasn't able to join live today, that'll become available to you usually about 24 hours after the conclusion of the webinar today. So look out for that. You should get that in your email. And without further ado, I will hand it over to Mitchell.
Thanks, McCain. So first of all, thanks, McCain, everybody from Leicester Media and DEI for having us on today. Again, my name is Mitch Feeney. I'm with Planet Earth Agronomy, and I want to throw out a little bit of an incentive to stay around till the end and participate in the live question and answer. We are going to do a raffle for everybody who sticks around to the end and asks some questions. It'll be 100 acres of free product, which constitutes about $1,000 worth of product. So if anyone's time is worth more than $1,000 an hour, please feel free to get out of here. But if it's not, stick around to the end and ask some good questions. I hope we have a little bit of a conversation at the end. So again, we're going to talk a little bit about modern farming and microbiology, and we're going to really focus on residue. Fred Bulow, and some of the more recent presentations I've been giving with him, he talks about it as it's really nature's biological residue is. And so we're going to talk about that in our presentation today. First, I want to start with a story. I was sitting on a plane with a Nebraska farmer, and we had 2 hours. We're going to talk about stuff, and he asked the question, why is everybody talking about biologicals? They've been around for thousands of years since life was kind of starting. What is the whole point of biologicals? Why is everybody talking about them? And we have a saying that we say usually when somebody screws up in the warehouse, but it's called trust but verify. And so we looked up on Google Trends, well, are people really talking about soil biologicals? Are they really talking about soil health? And the trends are showing that they really are. And hopefully that's because of our company and companies like ours that are out in the country at plot tours and at plot days, giving presentations on the value of soil biology and the value of soil health. But really, we are seeing that as the trend. The trend is going up. There's more conversations. It's trending toward people looking at soil biology as a really important factor on their farms. So then we look at the market as a whole. It's growing at about a little under 14% per year. That, when you compare it to, say, a fungicide market that's been around for 20, 25 years, that's growing at 6% per year. And it's expected that that biological market is going to reach about the same, at least projected revenue, as the fungicidal market by 2027, 2028 time period. So that's really exciting to see. Those who are using these products, they're actually getting a benefit from them. They're buying them year after year, and we're seeing kind of this growth. So again, why is that happening? And we're going to answer that question through the presentation today. Why are people using biologicals? The first way to answer that is really it comes from the microbiology segment. So it comes from, for example, Dr. James White from Rutgers University, who has been working with Australian scientists who discovered this process called rhizophagy about 17 years ago. They discovered this process that basically explains what soil microbes are doing in the soil, how they're being absorbed into the plant, and then how they're being ejected out the root system and then regrown. They don't just die when they're absorbed into the plant and they're never used again. The good stuff like the DNA, the organelles, those pieces and components are still alive and ejected back out into the soil profile. And it kind of explains that 8th grade biology term, that circle of life. Those soil microbes still exist. So this is a video that they took, a little movie that they took that we turned into a GIF, showing those soil microbes ejecting out of a root tip and being regrown. And the process, that rhizophagy process that we're going to talk about a little bit, is the process of those microbes reforming at the root tip. The root tip will feed them synthate. They'll regrow and they'll kind of balloon up. Then they'll be sent out and tethered back into the soil profile, and they'll go back to doing that good in your soil. They'll keep mining nutrients off of the soil colloids. They'll continue to break down residue, and they'll continue that cycle until they come back into the root system and bring that nutrition back into the plant. So that process is reformation. That process is the plant fills the microbe with root exudates or feeds it root exudates. It enlarges enough to detach from the root tip, and then it goes and captures the soil nutrients and kind of mines them off the soil colloids. And then again, being sucked back into the root system, and that cycle starts over again. Dr. James White calls that process and coined that process rhizophagy, which is just this big, really term for the soil microbes life cycle. So it starts, the reason why we talk about biologicals, with a better understanding for soil biologicals. When I started in this business, we would run around. I ride with my dad in the truck, and he'd give me a bug net, and he'd say, "Go count bugs." We'd test those bugs, and we'd count those bugs, and we'd try to come up with a really good way to evaluate what was going on. Well, that really comes to fruition with soil biology as well. If we don't have a way of testing for it and counting and coming up with a way to evaluate these products, we don't really have a good understanding of how they work. So about 10 years ago, Sylvita came out with a CO2 burst test. This is like the COVID test for soil, right? You stick it in the soil, you wait 24 hours, and the soil will respire or let off CO2. And this little piece that you put in the soil will change color based on how many soil microbes and how active those soil microbes are in the soil. And so this was a really basic test, and it kind of changed the game and allowed us to really evaluate how soils were respiring, whether soils were alive or dead, and what was kind of going on in the soil. The problem with these tests was we really couldn't compare them day to day, week to week, different types of soil, different parts of the field. We weren't getting a number that we could kind of associate if we were building soil biology. We were just getting this kind of yes or no, is the soil alive? These still exist today. We still use them all the time as a really quick assessment. In 24 hours, I know whether I have good soil biology or bad soil biology, depending on whether this comes back with some respiration, or at least I know that there's something alive in my soil doing something. These tests have gotten a lot better, though. So Dr. Haney came out with the Haney Test. That's a much better, all-encompassing test. You send your soil in, for example, to Midwest Labs, or really any lab now, and they can run this Haney Test. It starts with that CO2 burst test. It gives us an evaluation, is the soil respiring? But it also takes into account organic carbon, organic nitrogen, the ratio between organic carbon and nitrogen, and then gives us this soil health calculation. So as you're sending in soil samples, as you're learning a little bit more about this process, I really, check that box for, I'd like to learn a little bit more about my soil health, and they'll send you back these reports when you're sending in your soil tests. Most of the farms we are on sit right between 4 and 7 for the soil health calculation. So that to me means we really have a lot of work to do on our soil health. And it makes sense. We didn't have the tools to improve soil health until much more recently. And there weren't a lot of farmers like yourselves out there doing no-till practices. So they were doing practices that were a little bit hard on the soil, and they were breaking it apart and destroying the fungi and really breaking this down. And that's why we see most of the farms we go on kind of between about a 4 and a 7 for that soil health calculation. I was in Texas, and we did pull some soil there that was about zeroed out on soil health. And we have a lot of work to do to kind of build their soil health back. But we have seen that low. We didn't think we could zero it out, but there are soils in the United States that have pretty much next to zero soil biology in them. And pretty much nothing grows on those soils. Nothing can grow in an environment where there is no soil health. Taking it a step further, now we have DNA sampling. So most guys, they probably never heard of this, but some of you probably have because you're into this. We can test the soil for the specific microbes that do specific things. So nitrogen fixers, phosphorus solubilizers, potassium cyclers. We can test the soil and actually run a DNA analysis and learn a lot more about the soil than we previously did. The problem with them is they're very expensive. When we sent our first sample in for DNA testing, it was $2,000. And it was one soil sample being sent in. So they're not realistic on a larger scale. That's why we really like the Haney test, because you can send your normal soil sampling in, get a soil health calculation, and start understanding where you're at from a soil health standpoint. And then compare it year after year. Are we building more soil biology? Are we building more microbes as we go? So again, that goes back to that question, why soil biologicals and why are biologicals getting better? Well, because we can test for them and we understand them better. This was a field that we were on, similar to that kind of story about Texas, where they just couldn't grow soybeans. They were growing like 20, 25 bushel beans, and they were wondering what was going on. It was a newer farm that they had purchased. They were trying to figure out more of the history on the farm. And we went out and we did these soil health tests, and we realized there's basically no biology in it. Kind of go down the road about a month, we learn a little bit more about the history of the farm. We realized that this farm was basically, they didn't plant a crop in it for about 2 years while they were selling it. And they just burned down the weeds in it and they kind of left it barren. And the problem with doing that is you get this fallow ground syndrome, which is soil microbes really need plants to survive, and vice versa. Plants really need soil biologicals or biology in the soil to survive. If you kill them both and you run them down, you leave your soil barren for long enough, you'll break down that soil biology to just about nothing, and then it's going to be really hard to grow a crop on it again. So, it wasn't a great one to inherit, and it wasn't a great problem to work with. But we were able to go through and put some soil biology back on in these, it's a couple rows here just to prove, hey, some of this could work. There was no fertilizer in that application whatsoever. It was just bacillus microbes. So just bacteria to go in there and start moving that fertility that's trapped on the soil already with those soil microbes. So that was like in 2010, where these products really didn't exist. They were really difficult to use, but we got visual results back in 2010 with some of this stuff. Furthermore, why are biologicals becoming more and more prevalent? We're learning a lot more about them. We're starting to see these visual results. So we did a volunteer corn trial where we were just looking at how much volunteer corn is going to pop up in treated and untreated. And what we're learning is when we put on a product like Reclaim, which is a residue breakdown product, it has the tendency to cause that volunteer corn to germinate in the fall during that fall application and then freeze off, at least here in the northern states. So we don't have as much volunteer corn. It's germinating early. Therefore, we don't have it come spring when we're going in and planting beans, for example. So we're starting to see, this feedback, and I love seeing this feedback from our growers. They're sending us messages and emails. Hey, have you seen this yet? Have you seen that yet? And we're seeing a lot of these kind of side-by-side pictures. This is treated and untreated with Reclaim, showing right after the planter goes through. And the reason I show this is because I think a lot of people are looking for like this magic bullet, this amazing product that you put out and you can break down 100% of the residue that's out in the field. That really doesn't exist, right? We're still going to have that outer shell of the residue. The lignin out there is really hard to break down. But what happens when we put these products, these soil microbes out there, like what's in Reclaim, we're eating the pith out. So that center photo there is showing on the left-hand side, the pith is being eaten out. And the problem with that pith when you take your planter through it is it's like a rope. So you're going to pin trash and you're going to plug things up. You're going to have a poor stand when you have residue that's not breaking down properly. And when you put on soil biology and you put those residue breakdown products on, like Reclaim, we're able to break down the pith. We're able to kind of turn that rope into something brittle, and the coulter cuts right through. So you see this picture on the left where the planter's working properly, the trash rippers are working properly, and on the right, the untreated, these are both planted fields. Your equipment isn't working near as well, and so you're going to have uneven stand as we go through there. And then as we continue to answer this why question, why are biologicals, finally coming to the market? Well, there was a lot of work to do. 17 years ago, when they discovered rhizophagy, microbes could not be mixed with, city water. Nonetheless, a herbicide in your burn down applications. They just weren't functional. They couldn't be put out on a crop during our current cropping rotations because they just didn't survive the tank mixes. So what we've focused on is we've taken that soil biology that's native to the soil and we've made it durable. So we've taken a crop-specific consortiums that are built for corn, built for residue, built for soybeans, and we've designed them to have really high CFU counts, so high colony forming units, so we can really get out there at high populations and make a difference. We can make sure that they survive going through a pump with well water, chlorinated water, all kinds of different mixtures and compatibilities with nutrients and crop protection products. We want to make sure that they survive and get out into the field. And that's been our core focus. So there are products out there that are still like the 20-year-old technology that don't survive, just going through a pump, let alone being mixed with a herbicide application, which a lot of this product is going out as a herbicide or as a burn down application. Long shelf life is also super important to make it functional for our industry. You got to have something that's going to carry over. A lot of the products on the market, today and over the last 10 years, they have to be used in the single season, and they're manufactured, a week or 2 or 3 before they go out and they get sprayed. Anything after that, you lose your colony forming units and you lose the efficacy of the product. So, for example, Reclaim, it's got a 2-year shelf life in liquid form. Our dry products that we sell have, a 5-year shelf life. So we're starting to get these longer shelf lives out of the product by making these biologicals more durable. And then lastly, no fermentation. A lot of these products require you to put them in the mix and wait 24 to 48 hours for them to start the fermentation process. You will not need to ferment any of our products. And all of those things create consistent performance. So we won't get consistent performance if we don't have a combination of all these things. We're going to get a year where it works and a year where it doesn't. We're going to get throughout the country and different soil types and different types of operations where they're using city water where it won't work in some applications. And that's why our microbes have the most consistently across the field. We've got plot data that shows it. We've got all kinds of farmers using it now, and we're on now millions of acres with these products because we get consistent performance and consistent results with them. That's not to say that it's going to work 100% of the time, right? And we do get feedback from growers, and we want to make sure that we get that feedback because we want to go out and find out why these products don't work in the small cases where they don't. So once in a while, like once a year, we go out and we check out a field that is just it's an anomaly, and we try to fix those problems too for those growers that are having problems with products. So we're not saying it's going to work every single year 100% of the time, but we have created the most consistent product line that's on the market, and I'm confident in saying that. Another reason why we're starting to get a lot more buzz around biologicals and soil biology is Dr. Fred Bulow, who's kind of, a big deal in the industry, is this 7 wonders of corn. And he actually revisited this, and he, went from saying that number 7 growth regulators, I'm going to change that and rephrase that to biologicals, because really biologicals can cause a 10 bushel per acre return in my 7 wonders of corn. And in addition to that, it's the only one, this is a quote from Fred, it's the only one that you can do more than once. It's the only one that has versatility. Biologicals are incredibly versatile. They can be applied for nutrient release applications. They can be applied on seed treatments. They can be impregnated on dry fertilizer. They're placed in starters. They're used in biocontrol applications. And like today we're going to talk about is residue management applications. But they're so versatile, And there is an opportunity to get more than 10 bushels from these product lines because you can put them on more than once throughout the season and put them on for different reasons. And so I'm excited to see that, we're seeing, kind of a revisiting of the 7 wonders of corn. And he's been going around and kind of the speaking circuit with us in the various plots, talking about biologicals and why they're important and why they're important to incorporate into this research. So again, to rephrase, why is everybody talking about biologicals? Well, we understand them better. We can test them. We've made them functional. We can see visual results. The market is growing and money is being invested, and biologicals are versatile and they have a compounding effect. And then I threw this 7th kind of one in here. I don't like to talk about this all the time, but we do have outside pressures too. There are areas of the country where we're being limited on the amount of nitrogen that we can use. So we can put out 170 units of nitrogen, 175 units of nitrogen, and that's the most we can put out. How are we going to get 200 to 300 bushel corn in those environments where we're pulling back on nitrogen? We need to be more efficient with our nitrogen usage, and the way we can be more efficient with our nitrogen usage is to looking at these biologicals and looking at soil health. So we're going to go into now what residue management practices we can do and what those entail. We're going to talk about Reclaim, the product from DEI that specifically is for breaking down residue, improving soil health, and starting that nutrient cycling at the right time for your crop. And so we'll talk a little bit about crop residue next. So the conversation I had with Fred recently was, is crop residue an adversary or an ally, friend or foe? And we talk a lot about how it's an adversary. A lot of people will come up to us in trade show booths and we'll be talking and having discussion around crop residue, and they call it trash, right? They call it, it's in the way, it's causing problems. It plugs planters, it causes uneven emergence, it traps nutrients like nitrogen, it wears our equipment down. I had a grower tell me he spent $10,000 a year on tires. So he's worried about that's caused by the residue. He's running it over, it's ripping his tires apart. And it looks ugly, right? It's not a visually appealing thing. People don't like seeing that uneven emergence. They don't like how it looks in the field. We really need to change that. I'm hoping that over time people look at residue and they smile and they look at it and say, residue is a good thing. It's a good thing for our farm. It's helping us. It's doing a really good thing. And the way we can talk about that is we talk about residue as an ally, but it's only an ally if we manage it appropriately. If we don't manage our allies, they become adversaries very quickly, right? If we don't have conversations with our allies, they go rogue and they cause problems for us. Well, with crop residue, it's no different. If we don't manage it, it becomes an adversary, and all of those problems can happen. But if we manage it effectively, it is an ally. It builds soil organic matter. It's able to regulate soil moisture and temperature, keeps our soil moisture and doesn't let it drain away. It allows us to prevent soil erosion. It's food for microorganisms that kind of keep that cycle of life going and pulls nutrients off the soil and feeds them into your crop. And they're a massive source of mineral nutrients. And we're going to talk about that next. So University of Illinois did a long-term study on how much nutritional value does your residue have in it. And so this is corn. This is an example on corn. The value of residue, pounds per ton of residue. And the typical field that we go in, if we were to weigh all the residue in the field on an acreage basis, we're going to see on average about 3 to 4 tons per acre of residue out in the field. How much nutritional value is in that? Well, right here, it kind of explains what that looks like. So there's 20 pounds of N, there's 4 pounds of phosphorus, and then there's 69 pounds of potassium. So some potash is out there. And what's really cool about potash is, and Fred talks about this on his circuit too, the cations like potassium in the residue is 2 to 3 times more available to the plant than chemical fertilizers, than potash. So you can get a lot more value out of your residue from a potassium standpoint than you can by putting potash out. So that was really cool to hear and see the research behind it and show people, hey, there's a lot of value in those residues. So there's places in the country where they literally burn their residue off. let's stop burning residue, right? That's a very important thing to keep and hold and just manage it a little bit more effectively. If you're in an area where you have 200 bushel yields, again, this is for corn, 200 bushel yields, you have about 90 pounds of N that's trapped in that residue. If you're at 300 bushel corn, you have about 136 pounds of N. So we're looking at this and saying, depending on how much you're yielding, that's how much nutritional value is in that corn stalk. So let's try to release that at the right time during the growing season so we can get that to return to the plant and get it reabsorbed in the plant and recycle it. So we're not spending as much. I mean, with fertilizer prices where they've been the last couple of years, I think everybody would like a little bit of free nutritional value. So again, let's talk about this is just one of the approaches for managing residue. I mean, and I want to just briefly talk about when we harvest, one of the most important things to do is try to get even coverage of our crop residue. Try not to have it in, piles, because then you're going to get streaking in the field. So there's a lot of things we can do, not even tillage practices, but we can do when we harvest to try and maintain good coverage of our residue. So there's a couple things there, and then we can use management practices like putting reclaim on. And so this is an example of a bin study that we did. This is just to show you kind of the active biology and how it looks and how it functions. So we have treated on the left, treated on the right. And what we're doing here is we're trying to measure how much nutritional release. Again, trust but verify. We saw Fred's presentation. We wanted to verify, is that really happening out in the field? Is that really happening with our residue itself? So we basically stacked 2 bins, drilled some holes in these bins and let the water kind of wash through them. We treated the section on the right, and you can see after 3 days, we're already seeing that soil microbes go to work. They're growing and they're bringing that fungi in, and they're starting to break down the pith in that residue. What these microbes do when they're applied, once they hit soil or once they hit a food source, they're going to start doubling in population every 45 minutes to about an hour and a half, depending on the soil temperature. They will absolutely slow down when soil is cold, and they'll absolutely speed up when the soil is warm. But they're doubling in population, so there's a geometric effect. So if we're putting billions of microbes on, we're able to have this doubling effect, we can get a lot of work done in a short period of time. When we first were testing this product line, we thought it was going to take at a minimum of 60 days to see any sort of difference. And we were really astonished at how quickly these microbes will double and will get up to their populations and will start doing their work. That said, right, we deal at least I'm from Wisconsin, so we're in the northern part of the country. We have a lot of snow and issues that we have to work with on cold. It's not going to happen in 3 days here in Wisconsin. It's going to be a much slower process here to break down that residue. Reclaim itself. So again, we're talking about that doubling effect. The microbial consortium that's in there is 8 different microbes. So there's 8 different bacillus species. So these are bacteria species that are going to go in and they're going to start eating out the pith. And then there's a carrier on there that helps house those bacillus, protect that bacillus, keep it alive, keep it happy and healthy. There's a food source in there that helps, and it's like it's more of a biostimulant effect that we're getting from that product. It's helping stimulate those microbes growing and stimulate the fungi to start breaking down that residue. And that, I think, is really the key. Is the fungi is going to do the heavy lifting. The bacillus will feed the fungi nutrients to keep them going to break down that residue quicker and faster and more uniformly. So Reclaim is the product we're talking about. It's a crop residue decomposition product. It does nutrient release. And then we'll talk about in a minute, we're improving soil health with this product as well. It goes on just to talk about use rate. It's a pretty easy product to use, 12.8 ounces per acre. So we're able to apply it, a gallon treats 10 acres. So it's pretty easy when you're doing your math for going out and treating products. This is the results of that bin study. So that side-by-side bin study that I was showing you before, what we did is we trapped all the nutrients in a closed system, and we used a resin ball, which basically mimics a root system and absorbs the nutrients in. And we did this at a 15-day and a 30-day mark. So you can see, my last name, untreated 15 days, treated 15 days, untreated 30, and treated 30. And what we were really surprised about, and then when we talked to Dr. White, it wasn't so surprising, is the total N was like amazing. And it was pretty much all in ammonium form. So you see here, you go from 8 to 240 parts per million in ammonium nitrogen. That was surprising to us because we were sitting there and we're saying there's not that much nitrogen in those corn stalks. It's mostly carbon in those corn stalks. There's not going to be that much nitrogen there. And Dr. White explained to us, well, that is the nitrogen fixing effect from those microbes. Those microbes need to be feeding the fungi, and those fungi love nitrogen. So they're going to feed the fungi nitrogen by fixing it from the air. So in this closed system, we were actually getting nitrogen fixation in the bottom section of that little bin, and we're seeing that nitrogen release as we're going. Some of that nitrogen is coming from being trapped in the stalks. Some of that nitrogen is coming from that breakdown process where those microbes are going in there and they're fixing nitrogen from the air. Same thing, P and K, the P and K numbers, P is pretty aggressive, but the K numbers aren't as aggressive. And that makes sense because K is going to be harder to break down and extract from that residue. So we went from 200 parts per million to 300 parts per million in 15 days. And in 30 days, we had 178 in the untreated parts per million and 564 in the treated. So we can get nutrient release fairly quickly on residue. Again, this is a closed system. So what does it look like when we actually get out and do a soil sample and look at what's happening in the soil? So we've got lots of soil health report data that's come back. So we've got with is in the blue, without here is in the orange. And again, you can kind of see the same story here. So, our potassium, magnesium, and calcium levels, you can see with the reclaimed product, 125 parts per million versus 106 without, 264 with for magnesium, 223 without, and you can see all these numbers. They're just boosting them up a little bit. And this wasn't, this wasn't a very long time. This was 60 days post application, and we're already starting to see that nutrient release in the actual soil samples, not just in the residue samples or in a closed system. Some interesting things that we kind of looked at here too is, like in the treated versus untreated, there was a Lyme application in the untreated. And we asked our question, why is that happening? And we're doing some additional research this year to find out why is that pH of the soil bumping up slightly in the treated area versus the untreated area. And when we look at our base saturation levels, it may be because our mag and cal levels are going up. So those are positively charged ions. So when those go up, we are going to improve the soil pH a little bit. We're going to bump it up and bring it closer to that kind of balanced state of 7. But we're still learning more about that. And then it was interesting to see the hydrogen numbers. The hydrogen numbers in both the treated segments zeroed out completely. And so again, we're learning more about what that means and how that helps. But hydrogen is really an element that it's not helping us, right? It's not something that the plant can use or utilize. It has to convert it into water and transpire it off. It has to use energy when you talk about how it's going to utilize or use hydrogen. Whereas cal and mag, those are actually showing up in the soil pH. So we're going to learn a little bit more about what that means and how we can continue that process. But we're seeing it in both the closed system, treating the residue and showing how much the residue is releasing, but we're also seeing it in our soil sampling. So then that leads to the next step is, what trust but verify, how much residue can we really remove? How much good can we really do? So the first study that was done on this, where we sent some product down, was in Auburn. Auburn did this study, and it was an awesome opportunity to really look at how much residue we can break down with these various ways of doing it. My dad and I talk about, hey, 20 years ago, people talked about putting UAN on to break down residue. And some guys say it works great. Others say, it doesn't work at all. Well, they put 6% UAN on, and they were able to see about 18% on GMO corn, 18% by weight breakdown compared to the untreated control, which was 6.5%. So in untreated residue, on residue we just leave out there in a no-till environment, if we do absolutely nothing to it and we don't have a product out there to help break down residue, we're breaking down about 6% by weight in a year basis from fall to spring. So that shows we are breaking some residue down, and if we do improve soil biology, this number will improve, but we can do some good by putting UAN on. So we are getting like 3 times the breakdown when we look at putting UAN on, but not good enough. Again, the feedback that we get from the country is, hey, some guys are saying it really doesn't break the residue down at all. Humic acid does a little bit better than UAN at 22.5% by weight breakdown. The residue bacterias that are used do about 34%. So these just straight bacteria products will do about 34% breakdown, and Reclaim at the current use rate that we're using today is 51.6% breakdown. So about half the residue from the time that they did this. Basically they took a square meter and cut out all the residue, bagged it up and weighed it. 50% by weight of that residue was gone post application. And so we can do a lot of good, and we can actually make a difference now. If half of the residue by weight is gone, that's making a substantial difference, and it's releasing a lot of nutrition back into our soil. So we have testing, and I can send this data out later. This will obviously be recorded, but we have data on soybeans, on hay, on wheat straw. So we do have other crops, and we're getting very similar data. About half of the residue by weight is getting broken down in an accelerated manner. Again, trust but verify. We've replicated this trial in a couple areas of the country, because when we first saw this data, yeah, that's great, it looks awesome, but we're here in Wisconsin. Our growers are in a totally different environment. So we replicated in Black Earth, Wisconsin. We hired a third-party agronomist to do the study and just said, hey, replicate the Auburn study and see what we get. She weighed all the untreated and treated trials. So this is kind of some of the raw data. I've got a little YouTube link there showing her cutting the square meter areas. And then she looked at it and said, well, how much residue did we break down? And it was almost exactly the same, about half of the residue. So there was 7,437 pounds of residue in the untreated section, and there was 4,136 pounds of residue in the treated section. So about half the residue was broken down between the treated and the untreated. And then she went the step further and took Fred Bulow's research and replicated that. And again, this is about a year or two old now. So these prices are going to need to be relooked at. But there was about $70 in 1 acre of nutrients released from breaking down half of those corn stalks. So there's 28 pounds of urea, 7 pounds of DAP, 56 pounds of potash. And she took that and put it out in kind of a cost per acreage. So there was actually a pretty good nutrient release. And what I like about this is Reclaim can allow us to release those nutrients at the right time, rather than having it be released in the wintertime where some of that's going to flush through. We can release this kind of right in the spring season and kind of blow that up and get the value out of releasing the nitrogen and the potash applications at the right time. And she didn't take and multiply it by 3, but the potash applications, according to Fred, are 2 to 3 times more available than a potash application that's not coming from residue. This is just a side-by-side picture. I love this picture because it really does show, she's got all of it bagged up in the treated section on the left hand and then the untreated on the right. And there's, you can visually see how much more trash there was in the field. This, trial, we were doing 3 years no-till. And so we've continued to use this product year after year, and we're seeing, pretty substantial over time. We're getting better and better yields. I don't think it'll continue forever. We're not going to just see, this continued linear effect every year we use it. But the first year we put this on, we saw about a 5 or 6 bushel advantage from the treated and untreated from using Reclaim. We think most of that probably came from stand. We just had a much better stand in the spring. We had more plants survive and come out of the ground, and we had it was a nice picket fence coming through. After this 2 and 3 years, we've increased the amount of bushel advantage, or the yield advantage that we've got. So in the untreated check, we had 152 bushels. This was a tough year for us. We had a frost event happen that froze off a lot of the corn, and it did grow back. We got it to yield, but we saw a pretty drastic increase between the treated and untreated. 175 bushel on the treated segment here for 3 years no-till. Again, some more pictures of untreated and treated. This was that first year, or this was the first application in this particular area that was being treated, and we had about a 5 bushel advantage over the check. When we mixed a biostimulant with this product, we ended up with an 11 bushel advantage. So these biostimulants and these microbial products work really well together. So think the microalgae and the seaweed products. If you can mix the seaweed products and microalgae when you're going out to spray, you can actually get a kind of a doubling benefit from that. They work together really well. They stimulate the microbes. The microbes love microalgae, and they'll grow even faster. Now, Reclaim already has a biostimulant incorporated into the product that's shelf stable. You have to be really careful with mixing Reclaim with a biostimulant and then leaving it sit for a long period of time. If you leave it for 2-3 weeks, your jugs will start blowing up. So don't pre-make a bunch of this for the season. If you're mixing a biostimulant along with Reclaim, mix it up and go spray it fairly soon. You got 3 days or so to spray it before you'll start getting activity, but they really will start moving if you do that. When we see emergence coming out, this is what we see. We get lots of pictures from growers using this product, and we see these kind of side by side where you just don't get even stand. You don't get as good a plant health. You don't have the equipment working properly when it's going through. The coulters aren't working. They're not cutting through the trash, and so you're not getting even emergence. Whereas when we had a fall applied application of Reclaim, really nice uniform stand. We're trapping kind of all that soil moisture in the residue rather than letting it, go off. And we're seeing a really good advantage from a plant stand and a plant health standpoint. And I think a lot of that is from this third picture over on the right. Those microbes love roots. And so they are eating and tearing apart those root balls. And so we're not seeing kind of that plugging effect or that pinning effect that happens from pinning those root balls as we're going across. They're breaking them right apart. More pictures of treated, untreated later in the season. This was done in Whitewater, Wisconsin at the Agritech farm. So this is a research farm. And, just showing that as the season continues, we're seeing corn, a couple stages ahead, and quite a bit taller, quite a bit bigger leaf area index, and we're seeing a pretty good advantage. And the fall Reclaim application in Whitewater had about a 12 bushel advantage over the check when the data came back for this one. Our best return on investment is soybeans planted into corn stubble, and that's probably not super surprising. You guys have all probably planted beans into corn stubble, and it looks like crap at the beginning of the year. It just doesn't look good. We don't get a good stand. We lose a lot of population, and we don't have good plant health early season. We see the biggest advantage from a yield standpoint in beans going into a no-till environment. You can see, these side-by-side pictures, the canopy is closing sooner. We see already less residue in between, and we have a really good environment there for our soybeans to grow up nice and healthy, pop out of the ground nice, and we don't have a lot of those stand issues earlier in the season. That being said, beans always seem to look like crap in the beginning of the season, and then they shine late season. So we get, pictures all the time. Hey, my beans don't look perfect. They look much better than the untreated check, but they don't look perfect. Beans just have a hard time looking good early season. And then late season, they come back and they really shine for us, especially this year, at least in our local region. The beans look phenomenal for how much drought we had early season. This is a grower that split a field. He's been using Reclaim. Now it's his 4th year of using Reclaim. And he called us. And farmers love calling and getting excited. He goes, you can see it from space. All right. Send this to everybody, you can see it from space. And we're kind of laughing. We're like, well, that's awesome. We're seeing this from space. So the satellite imagery is also showing us this improved biomass. So that purple segment, that's like the best biomass that you can get on the treated section of Reclaim versus the untreated section of Reclaim. And this is a farmer. He farms with his brother, Dave and Dale, and they are a blast to work with. They were one of the first people. They're an innovative grower, one of the first people to use this product when it was released, and they've been using it ever since. And they put it on most of their acres, but they did keep like a little segment, like 5 acres, untreated the last 4 years so that we could do this research. And so I got to thank growers like that who keep an untreated check and keep it consistently, because it helps everybody learn a lot more about how these products work over time, year after year. And it's really hard not to go out and spray it on everything when something is working. The last thing that I'm going to mention here is the soil health component. I've got a soil health report that I hand out at every one of our discussions. Please feel free to, there was I think a click for more information, and you'll get a digital copy of this. If you're at any of the grower events that we're at, we'll hand these out. This really summarizes a lot of what I went through, but targets the Auburn study, the replication that we had of the Auburn study, and it's an in-depth discussion about soil health. So what we've done over the years now is we've been sending these Haney tests in and summarizing them. And so our untreated check, and we did it partially, we did this research to find out what dosages are going to be the best. What is the best dose of Reclaim, the concentration of microbes that we're going to need to be able to get the best results on the soil health standpoint. And we thought, first year we put it out for the soil health report. Let's see if we don't care about residue breakdown. We know what we need for residue breakdown, but if we just care about putting something on for soil health, where can we bring the rate down to? So we put the rate down at half rate. We did a full rate. We did a 2 times rate. And what you don't see here is we also did a 10x and a 20x rate to see what we could do to soil health. And we wanted to see a couple of different things. Is if we lower our concentration of microbes, do we gain the benefit? If we bring our CFUs down, and will we get the benefit? And the answer is really no. These microbiologists, there's a reason why they've been studying this for the time that they have. They really know what to put on at what concentration to get the greatest benefit, so that those microbes go into an environment where they're going to be successful. If we don't put enough of them out there, they are unsuccessful. They can't compete in the soil, and they just don't work. And they might even have a backwards effect when we're not putting it in there on the soil health rating. This wasn't really statistically significant. We did have a little bit backward on half rate, but this just goes to show we really do need to put enough on. Don't be cutting rates of microbial products, because you're just not going to gain the benefit. You're not going to see the value of it. Vice versa, again, what I'm not showing here is there's a 10x and a 20x. You can also overwhelm the soil. And so if you do overwhelm the soil and you do too much, it'll do the exact same thing. If you put 20 times the microbe concentration, we don't see this soil health benefit. Those microbes are going in and they're not being successful again, because we're putting them at too high of a population. It's too crowded for them. So half rate, again, unsuccessful. Full rate went from a soil health calculation of 12 to just under 20. And when we doubled the rate of application for microbes, we were just under 25, which is the maximum that the Haney test tests for. So I was really excited to see the Haney test come back, and it's starting to evaluate it year after year. We are really building soil health over time. And I think that's really where the discussion is. I mean, this is a no-till farmer group. You guys have been building soil health for years if you've been no-tilling, because you've been putting nature's natural biological out there, which is residue. And so your guys' soil health might be even better. This was in black earth, so really good dirt. I mean, amazing soil. We typically see, again, between 4 and 7 on these soil health calculations. and we were able to, just our untreated check had about a 12. So it's really good soil. It's really good, opportunity to grow a good crop on that, but we can still improve really good soil and get it up there to, 2025 soil health ratings. And as these tests get better and cheaper, it'll be much more accessible. I think this soil health report, each one of the tests that we send in are between 20 and 50 bucks, depending on how much we do with Midwest in the season. That's about what they're charging us. So they're becoming pretty cheap, and we're able to get some pretty good benefits from them. So without talking anymore, if you want to learn more about Reclaim and the product, please go to DEI-directenterprises.com. If you have questions, I'm happy to take them now. Again, there's my email, there's the Direct Enterprises website. Feel free to reach out with questions if you don't get it answered here during the live Q&A.
All right, thanks a lot, Mitch, and thanks everyone for being here. And we'll get to some questions here. I think we got about 10 or so minutes, and we'll get through as many as we can. So I'll go ahead and start with this one here, Mitch. We have Ed asking, what is the 1-day CO2 C burst? How does it relate to if one measured directly CO2 respiration rates?
Yeah, so what is the 1-day CO2 burst carbon burst rate? So that is the Sylvita test that you're referring to, I'm assuming. So what we're doing there is it's similar to like a COVID test. It's really just telling us, is there respiration in the soil or is there not respiration in the soil? How does that relate to one if measured directly, CO2 respiration rates? It would be very similar. I mean, this little piece, they're going to be identical. The difference is the CO2 respiration rates that you're getting from Midwest Labs, for example, they're going to give you an actual indexed number between like 0 and 100 for respiration rates. The CO2 burst test is going to give you a color that just correlates to that respiration. So I like the Midwest Lab soil reports much better. I hope that helps answer your question, Ed.
All right, and then a follow-up, any tests or analyticals for plant root extradites, example, organic acids? If so, how are these interpreted?
Yeah, so we've asked Midwest Labs to start including that information on their test reports. So instead of just giving us the 0 to 25 soil health calculations, start giving us the levels of organic acids that they're seeing and observing, and start telling us a little bit more about that. At this point, we don't have enough data to be conclusive on it. We don't really understand it enough on these soil health reports and how we can use them yet, but we're starting to get those. So, kind of stay tuned for those pieces.
All right, and then we have Mike asking, is there a magic number to reach in CFU count to be an effective product? And how does Reclaim keep that number over 2 years shelf life?
Yeah, really good question. The answer is no. There is not a magic number, because soil biology, these microbes that are being used in these products, there's lots of different types of microbes being used. So on an individual basis, there may be a really good number for colony formings, but there's not an exact or a perfect number for CFUs. What I'll say is you want to be in the billions with a B. Okay, so we're putting on about 300 billion microbes when we're pushing those out there, and then they're doubling in population. You want to be in the billions. A lot of these products, when you look at them, they're in the millions. They might be putting 30 million or 40 million or 50 million CFUs out. You want to be in the billions with a B. So very high concentrations. And then the next question to ask is, are those microbes sporulated? And how do they sporulate them? Some companies won't answer that. I've got people on our team that, some of that is proprietary on how they go through the sporulation process. But when you create a hard endospore, that's how you keep the microbes in Reclaim alive. They go into an endospore form. So if you want to compare the size of the microbe, when you have a live microbe, call it the size under a microscope of a basketball. When we're looking at them under a microscope, they're like this big. When they turn into a hard endospore, they're put under heat and pressure, right? So they're heated and pressured down, and they basically, in their dying process, they'll shoot out a little piece that looks like a pocket comb. And that pocket comb is much smaller, thousands of times smaller than that giant basketball. Those hard endospores can survive in chlorinated water. They can survive, a lot of these products come from the oil and gas industry, and they're shot down in the fracking process. They can survive in ridiculously harsh environments. And that technology, that fermentation and then sporulation process, is what keeps the microbes in Reclaim alive. And in addition to that, the liquid formulation that we sell also has humic acid in it that is like a house for microbes. So once they get out into the soil and they're in the soil and starting to come back alive again and starting to multiply, they have a nice humic acid product to be in that houses them and keeps them safe as they're going. Good question.
All right, then is the N2 fixation pathway N2 to NO3 to NH4? Overall, there are plenty of intermediates, including NO2 and others.
Yeah. So, I mean, my short answer, without dipping into the microbiology background, is there a pathway? Yes, there is a pathway. The long answer to that process, and Ed, I'd love to get in touch with you. I'd like to get you in touch with Dr. White, because I think you could go back and forth a lot on the fixation pathway and speak to it. But I don't have a great answer for what you're referring to here.
All right, we'll keep it moving, try to get as many of these as we can. So how did the recovered or residual N compare in the closed system versus the field test?
Yeah. So very similar. If you put them on a graph, they're about the same. So we're seeing the closed system versus the soil tests about the same as far as how much they're improving. But like you'd expect, in the closed system, we're capturing all that N, versus in our soil test, we're only capturing what's showing up in the soil test. So the numbers are much smaller in the soil example than they are in the closed system.
All right, how about, are there any pest management implications, positive, negative, or neutral, after treatment for the next crop rotation?
Yeah, we haven't. So from a fungal pathogen standpoint, there is, and we can't talk about this as a fungicide, right? That gets us in trouble. But there's pretty good knowledge on improving soil health. And when you put bacillus out, it has the tendency to take fungi and put it from a pathogenic or parasitic phase into a plant health phase or a residue breakdown phase. And that's really what we're doing with this fungi, is it's in a non-pathogenic phase of its life cycle. So we see, when we do these tests, we see reduced fusarium, we see reduced fungal pathogens when we do the DNA soil tests. That being said, this product is by no means a fungicide. So we are not claiming any pesticidal or fungicidal effects.
All right. Is it possible to restore the stocks of soil carbon by using no-till and crop residues, as well as cover crops, for the whole soil profile without using perennial herbaceous crops in the crop rotation?
Yeah, that's a great question. I mean, I think we're getting there. I think the answer is yes. We are getting to that point where we can store soil carbon. We have met a lot. I mean, I think everybody's kind of heard the buzzwords about carbon credits and about this whole process. When we're testing it, we're seeing that we are sequestering some carbon in the soil. We're breaking that organic matter down. We are seeing improvement there. It's inconsequential, though. I mean, we aren't seeing people getting paid carbon credits based on the soil tests that we're getting. I'd love to be able to get somebody who's out there that wants to do a long-term study that would look at how much carbon we are sequestering over time.
Is this product available for purchase in Canada?
That's a DEI question. We do work with some folks out in Canada. So at a limited rate, yes, we can export. We import lots of products from Italy and all over the world, Australia. So we do work outside of the United States. But yeah, if DEI will ship some to Canada, absolutely.
All right. Oh, and then we had a follow-up from the last question. Boris was hoping somebody can send such publications that you were talking about over.
Boris, yeah, absolutely. we can absolutely send that. And then Karen, the full rate was 12.8 ounces of the product. The product from the Auburn study was placed at 2 quarts of liquid with the same concentration of microbes as we put out at the 12.8 ounces. So the finished product, 12.8 ounces per acre, is what's being placed out on the field.
All right, just got a couple more here. How does this work with cover crops like cereal rye?
Yeah, so we have a lot of people use this on cover crops because a lot of places like out in Maryland are now basically wanting them to plant green. And so they're putting this on actually after planting, after they've, burned down their cover crop. And so yes, this product will start breaking down the residue. What's important to note is this product is going to work on the inner pith of most of the plant material. So you're still going to see that cover crop out there. It's still going to be a little bit of a yield drag, but we do see some mitigation. We do see a little bit less yield drag from that piece.
All right, and then when CA and MG increase, does that tie up plant available P?
Not from what we are seeing. However, I think a person might say that it would, because we are, we're increasing the soil pH. We're taking positive cations that are out there. P is a negative ion. Are we tying some of that up? I'm not seeing a lot of evidence of it, but there may be some limited evidence of that. Yes. Does your bio-fertilizer affect methogens emitting methane? So I think let me ask, I guess. I think this is probably for like digesting and digestion processes. So like in manure pits and things, we do have lots of people that are using these in manure pits. And we do see a benefit from using this product. What I will say is we have better products in our portfolio that are specifically designed for that. And I would say, you're paying for humic acid and some other things in that reclaimed product that you probably don't need. But as an easy button, yes, you're going to see a benefit of breaking down and starting that process in manure pits.
All right, we'll go ahead and stop there. Thanks everyone for those great questions, and just want to thank everyone for attending today. Quick reminder, like I said at the beginning, the presentation and the audio, it'll all be made available on the No-Till Farmer website if you'd like to review it or show it to anyone else. And you should receive an email, I think within 48 hours of the conclusion today, so watch your email for that. And once again, on behalf of Direct Enterprises and No-Till Farmer, just want to thank you for joining us today. Please feel free, as Mitch said, to reach out if you have other questions that you didn't get answered today. And always feel free to reach out to us at No-Till Farmer if you have ideas for other digital demos and webinars that you'd like to see us do. So thanks a lot, and enjoy the rest of your day, everybody.